Good morning, students. Myself, Jincy, lecturer from Emmanuel Arthur College of Pharmacy. Today, we are discussing about the topic pharmaceutical emulsion. Emulsion. An emulsion is a biphasic liquid dosage form in which two immiscible liquids, one of which is dispersed as finite globules in the other. The particle size of globules range from 0.25 to 25 micrometer. An emulsifying agent and mechanical energy are needed to join the phases. Then types of emulsion. Based on dispersed phase, first type is oil in water type, which means oil droplets are dispersed in water. Then water in oil type emulsion, which means water droplets are dispersed in oil. Then multiple emulsion, which means water in oil in water type emulsion. First one is oil in water type emulsion. In these emulsions, the oil droplets are dispersed as globules in the water. For examples, oil in water type emulsions mean for both internal and external use are for external benzyl benzoate emulsion, example gamma benzene hexachloride emulsion, etc. Then internal classification, vitamin A in corn oil in water, liquid paraffin in water, griseofulvin in corn oil in water, etc. Then microemulsions. Third one, these emulsions are known to contain globules having size around 0.01 micrometer. Globules of such sizes are incapable of refracting light and thus are invisible to the naked eye. Then fine emulsions. These emulsions are known to have a milky appearance and contain globules of 0.25 to 25 micrometer. Then multiple emulsions, which means water in oil in water type emulsion. The emulsions are generally referred to as emulsion within emulsion and are titled as water in oil in water or oil in water type emulsion. For example, incorporation of water in oil type emulsion in an aqueous phase produces water in oil in water type emulsion. Then oil in water type emulsion. Water in oil type emulsions. In these emulsions, the water is dispersed as globules in the oil. For example, butter, salad dressings. Then the water is in the dispersed phase and oil is in the continuous phase in water type emulsion. Water in oil type emulsions. Usually, these emulsions are applied topically as lotions and creams. These emulsions also provide emollient action. Wool fat, resins, beeswax, synthetic compounds, and soaps formed from divalent bases are a few emulsifying agents used for preparing water in oil type emulsion. Then, based on the size of liquid droplets, 0.2 to 50 millimeter micrometer macro emulsions. Then 0.01 to 0.2 micrometer micro emulsions. Then 50 to 1000 nanometer nano emulsions. Then difference between the oil in water type and water in oil emulsion. Oil in water, water is the dispersed medium and the oil is the dispersed phase. Then water type oil type emulsion, oil is the dispersed medium and water is the dispersed phase. Then water soluble drugs are more quickly released from oil in water type emulsion. Water in oil soluble drugs are more quickly released from water in oil emulsion. They are preferred for formulations meant for internal use as bitter taste of oils can be masked. With water in oil, they are preferred for formulations meant for external use like creams. Then oil in water, they are non-greasy and easily removable from the skin surface. Then water in oil, they are greasy and not water washable. Then oil in water, they are used externally to provide cooling effect. Water in oil, they are used externally to prevent evaporation of moisture from the surface of skin. For example, cold cream. Oil in water emulsions give a positive conductivity test as a water is the external phase which is a good conductor of electricity. Water in oil emulsions do not give a positive conductivity test as oil is the external phase which is a poor conductor of electricity. Then advantages of emulsions. The inedible oils can easily be administered as edible emulsions. The obnoxious taste of oils can be concealed. The flavoring of aqueous phase of emulsion is possible. Then they are more rapidly absorbed than the solid dosage forms. Two incompatible ingredients can easily be incorporated with one in each phase. Then disadvantages of emulsions. The manufacturing process of stable emulsion required technical experts and calculations of primary emulsion formula. A measuring device is employed for administering emulsions. To ensure uniform dose of distribution, they are shaken prior to administration. In case they are not shaken well before use, dose accuracy decreases in comparison to solutions. The dispersion system may get affected if the emulsions are improperly stored. Then the emulsions are stored in a glass or plastic containers, thus are bulky, difficult to transport, and may break the containers. Cracking of emulsion may occur as a result of microbial contamination. These are the disadvantages of emulsion. Then pharmaceutical application of emulsion. Oral, rectal and topical administration of oils and oil soluble drugs. Then the unpleasant taste or odor can be masked by emulsification. The absorption and penetration of medicament are enhanced by emulsification. Intramuscular injections of water soluble drugs or vaccine to provide so release. The use of sterile stable IV emulsion contain fats, carbohydrates, and vitamins as a potential nutrition. These are the applications of pharmaceutical emulsion. Then wetting agents, anionic, cationic, non-ionic. Oil in water emulsion and water in oil emulsion. Finely divided solids includes bentonite, magnesium hydroxide, aluminum hydroxide, oil in water type emulsion. Then emulsifying agents. An emulsifying agent should possess the following characteristics. Stable emulsions can be produced by using emulsifying agents. These agents reduce the interfacial tension between the two phases and then separate the globules by creating a barrier at the interface. Surfactants are mainly used as emulsifying agents. The solubility of emulsifying agents determines the kind of emulsions to be prepared. By using water soluble hydrophilic emulsifying agents, where water is the continuous phase, an oil in water emulsion is prepared. In contrast, by using more oil soluble emulsifying agents, where oil is the continuous phase, water in oil type emulsion is prepared. The phenomenon in which any added substance modifies the solubility of emulsifying agent and change the emulsion type is known as phase inversion. Examples of emulsifying agents include diethylene glycol monostearate 4.7, sucrose diolate 7.1, and polyxylene laurel ether 9.5, sodium oleate 18.0, polyxylene stearate 18.8, potassium oleate 20.0. Then identification test for emulsion. First one, dilution test, staining test, conductivity measurement and fluorescence test. The hydrophilic lipophilic balance system of emulsifying agents. An HAB number represents the relative proportions of the lipophilic and hydrophilic parts of the molecule. High numbers indicate a hydrophilic molecule and produce an oil in water emulsion. Low numbers indicates a lipophilic molecule and produce a water in oil type emulsion. Oils and waxy materials have a required HLB number which helps in the selection of appropriate emulsifying agents when the formulation of emulsion is. Liquid paraffin, for example, has a required HLB value of 4 to obtain a water in oil type emulsion and 10.5 for an oil in water emulsion. Then preparation of emulsion. The methods commonly used to prepare emulsions can be divided into two categories: trituration method and bottle method. First one is trituration method. In this trituration method, this method consists of a dry gum method and wet gum method. First one, dry gum method. In this method, the oil is first triturated with gum with a little amount of water to form a primary emulsion. The trituration is continued till a characteristic clicking sound is heard and the white grain is formed. Once the primary emulsion is formed, the remaining quantity of water is slowly added to form the final emulsion. Then, wet gum method. As the name implies, in this method, first gum and water are triturated together to form a mucilage. The required quantity of oil is then added gradually in small proportions with through trituration to form the primary emulsion. Once the primary emulsion has been formed, remaining quantity of water is added to make the final emulsion. Then, bottle method. This method is employed for preparing emulsion containing a volatile and other non-viscous oils. Both dry gum and wet gum methods can be employed for the preparation. As volatile oils have a low viscosity as compared to fixed oils, they require comparatively large quantity of gum for emulsification. In this method, oils or water is first shaken thoroughly and vigorously with a calculated amount of gum. Once this has emulsified completely, the second liquid is then added at all once and the bottle is again shaken vigorously to form the primary emulsion. More of water is added in small portions with constant agitation after each addition to produce the final volume. Then dry gum method. Formula for primary emulsion of dry gum or continental method. This method is used when the emulsions are to be prepared by using dry gum emulsions that are particularly acacia. The amount of gum used to be decided based on the oil nature. The formula suitable for primary emulsions of different types of oils are given. Formulae for primary emulsions are types of emulsions, fixed oils, for examples, liquid paraffin, castor oil, cod liver oil, and olive oil and almond oil. Oil for gum 1 and water into volatile oil types of oils, turpentine oil, for example, turpentine oils, and oil of cubebs, sandalwood oil. Then types of oils, oleoresinous substances, for example, balsam of Peru. Then wet gum method. This method involves the same proportion of oils, gum, water. For primary emulsion is a dry gum method. In this method, acacia is triturated with water till a mucilage is obtained. To this mucilage, oil is added at intervals with a constant and rapid trituration, which is continued even when all of the oil has been added. The emulsion is completely following the usual procedure. In Vidgam method, two methods are there: large scale preparation and small scale preparation. First one, large scale preparation. The large scale preparation of an emulsion is similar to the small scale preparation. The steps involved are the oil phase and water phase. The oil phase means containing hydrophobic constituents and water phase means containing hydrophilic constituents are heated separately in large tanks. Then, if waxes are present, both the phases are heated above the highest melting point of any component present. Then, one phase is pumped into the tank containing other phase. Agitation is continued during this addition. The emulsion is cooled and homogenized. Then, the dispersed phase should be vigorously agitated so that very small globules are produced. Energy in large amount should be expanded for heating and mixing. Equipment suitable for continuous processing are used. Some of the equipments used for the emulsifications are ordinary agitators, mechanical mixers, a mechanical mixers which includes two propeller type mixers and turbine mixers. Then third one is colloid mills and ultrasonic devices. Then equipment for emulsion. We already told the mechanical stirrers and propylene type mixers, tur- turbine mixers, homogenizers and colloid mills, ultrasonic phase. Stability of emulsion. An emulsion is said to be stable if it remains as such after its preparation, that is, the dispersed globules are uniformly distributed throughout the dispersion medium during its storage. The emulsion should be chemically stable and there should not be any bacterial growth during its shelf life. Emulsion instability may either reversible or irreversible and manifest in the following ways. First one, cracking, which means irreversible instability, and flocculation, creaming, and phase inversion. First one is cracking. Cracking means the separation of two layers of dispersed and continuous phase due to the coalescence of dispersed phase globules which are difficult to redisperse by shaking. Cracking may occur due to the following reasons. First one, by addition of emulsifying agent of opposite type, then by decomposition or precipitation of emulsifying agent, and by addition of common solvent by microorganisms, change in temperature by creaming. Then flocculation. In flocculate state, the secondary interaction, which means van der Waals forces maintain the droplets at a defined distance of separation. Application of shearing stress to the formulation, which means shaking, will redisperse these droplets to form a homogeneous formulation. Although flocculation may stabilize the formulation, there is also possibility that the close location of droplets would enable droplets coalescence to occur if the mechanical proportion of the properties of the interfacial films are compromised. Then creaming. Creaming may be defined as the upward movement of dispersed globules. The upward movement of dispersed globules to form a thick layer at a surface of emulsion. Creaming is temporary phase because it can be redistributed by mild shaking or stirring to get again a homogeneous emulsion. Then fourth one is a phase inversion. Phase inversion means the change of one type of emulsion into other type, that is oil in water emulsion changes into water in oil type and vice versa. Due to the following reasons, phase inversion takes place: by the addition of an electrolyte, by changing the phase volume ratio, by temperature change, by changing the emulsifying agent. The phase inversion can be minimized by keeping concentration of dispersed phase between 30 to 60 percentage, storing the emulsion in a cool place and using a proper emulsifying agent in adequate concentration. Then the identification tests illustrate certain methods to differentiate between oil in water and water in oil type emulsions. Some of the popularly used methods are miscibility test with oil or water. The liquids miscible with the continuous phase of emulsion will also be miscible with the emulsion. Then conductivity measurements. Emulsions having aqueous continuous phases rather than oily continuous phase will conduct electricity rapidly. Then third one is staining test. Staining test. In this test, a blend of water soluble or an oil soluble dye with the emulsion is subjected to microscopic observation to determine whether or not the continuous phase or the dispersed droplets are colored. The dispersed droplets are colored. Okay, then it is oil and water type emulsion miscibility test miscible with water and not with oil. Water in oil type emulsion miscible with oil and not with water. Then staining test for incorporation of oil soluble dye. Macroscopic examination includes the color produced is more pale than that of the water in oil type emulsion. When water in oil type emulsion, the color produced is more intense than that of an oil in water type emulsion. Microscopic examination for oil in water type emulsion. Colored globules are observed on a colorless background. Then, water in oil type emulsion, colorless globules are observed against a colored background. Then, conductivity test, third one. The last one is a conductivity test for oil in water emulsion. Water, being the continuous phase, will conduct electricity throughout the system. Two electrodes, when placed in such a preparation with a battery and suitable light source connected in series, will cause the lamp to glow. Then, water in oil type emulsion conductivity test. A preparation in which the oil is the continuous phase will not conduct electricity. The lamp will not glow or will only flicker occasionally. Then, evaluation test for pharmaceutical emulsion. Determination of particle size and particle count. The emulsions are also evaluated for any changes in their average particle size or the size distribution of droplets. Processes like optical microscopy and Coulter counter method are employed to perform this operation. And second one is determination of viscosity. The emulsions that are stored for longer time periods are checked for their viscosity using viscometers. Then third one is determination of phase separation. It is a method for examining the emulsion stability by either observing visually or measuring the volumes of separated phases. Then the fourth one is determination of electrophoretic properties. Flocculation of emulsion can be evaluated by determining its electrophoretic properties like theta potential, since electrical charge on particle surface affects the flocculation rate. The following methods are used to evaluating the physical stability of pharmaceutical emulsion. Extent of phase separation. First one. The study of phase separation provides a proper understanding of practical and commercial features of stability. The study involves a quick method and is used to evaluate poorly formed and rapidly breaking emulsions. Even if the indicators of instability occur at any early stage, phase separation occurs after a certain time period. Then, second one is globule size distribution. The appearance of bigger size globules is an earlier sign of instability. The small globules aggregate and coalescence prolonged storage of emulsion, thus leading to its instability. The globule size distribution of emulsions can be analyzed microscopically for evaluating its physical stability. This method is almost similar to the uh, optical microscopy method for a particle size analysis in micrometers. Then, the surface area of globules is more associated with physical stability than the globule size distribution. After its manufacture, the emulsion immediately undergoes active coalescence for a time period throughout which the emulsion becomes stable and independent of the strains induced in the preparation. After this time period, the emulsion remains stable on extended storage. Then third one is accelerated stability studies, which means centrifugation. Flocculation and creaming are slow process which can be accelerated by using ultracentrifuge producing stress conditions within the system. At room temperature, the emulsions are exposed to diverse range of centrifugal speed and the phase separation is examined at certain time intervals. Then fourth one is microwave irradiation. In this irradiation, the emulsion is subjected to microwave radiations. Subsequently, its surface temperature increases and the temperature gradient existing between the surface and the bottom should be small enough to maintain the stability. Then the other stability problems and methods to overcome the pharmaceutical emulsion and breaking and cracking. This process involves both coalescence of the dispersed globules and separation of the dispersed phase. The merits of emulsification are lost since it is not possible to redistribute the globules and also precise dosage form. It is impossible. Then cause any physical, biological, or chemical effect that modifies the nature of the interfacial film of an emulsifying agent is known as that cause cracking. Following physical features are known to cause breaking. Opposite types of emulsifying agents: addition of common solvent, temperature, freezing, incorporation of excess dispersed phase. These are the causes of cracking, breaking of pharmaceutical emulsion. It's a cause stability problem. First one is opposite type of emulsifying agent. If an opposite type of emulsifying agent is added, emulsification reduces and cracking occurs. For example, soap sets of sodium lauryl sulfate produce an oil in water type emulsion, whereas soaps of calcium lauryl sulfate produce a water in oil emulsion. The process of adding monovalent soap emulsion to a divalent soap emulsion or vice versa can instabilize the emulsion and make them prone to cracking. Addition of common solvent. Second one is addition of common solvent. The solvent in which both disperse and continuous phase are soluble can be added to form a single phase system. Then the third one is temperature. Some of the proteinous macromolecular emulsifiers may coagulate at high temperatures and instabilize an emulsion. This is because the number of collisions between the globules increases with the increase in temperature. These increased numbers of collisions are known to overcome the barrier to emulsions, coalescence, and instability. Then, fourth one is freezing. The aqueous phase of an emulsion transforms into ice if stored below 4 degrees Celsius. The pressure applied by these ice crystals on the oil phase may initiate cracking. Then, fifth one is incorporation of excess dispersed phase. An emulsion is more liable to crack if the dispersed phase concentration is more than 74%. Inadequate addition of emulsifying agent in the preparation also leads to cracking or breaking. Then remedy. The following steps must be undertaken to overcome this difficulty. First one, more emulsifying agents should be added in the preparation. Then the temperature at which the emulsions are stored should be under the control. Then the concentration of dispersed phase should be regulated. Then adequate quantity of emulsifying agent should be added. Then phase inversion. The phases of an emulsion undergo inversion when its transforms from an oil in water emulsion to a water in oil emulsion and vice versa. Chemical degradation may be responsible for this phenomenon. However, the following physical factors are known to affect inversion: alteration in the phase volume ratio of emulsion and cooling those emulsion prepared by heating and mixing of two phases. Then, deterioration of microorganisms. The molds, yeast, bacteria may degrade the emulsifier, infect the aqueous phase, and degrade the oil soluble vitamins. Remedy. These problems can be overcome by adding adequate quantity of preservative in this preparation. Mixture of para-hydroxybenzoate and methyl ester and propyl ester are often used as a preservative. The preservatives with solubility in both the phases of emulsion should be used. If the preservative is not soluble in both the phases, continuous or dispersed phase, which means a suitable preservative for each oil an accused phase should be used. The preservative to be used should not form any complex with the emulsifying agent and other ingredients. Then, the last one of stability problem is miscellaneous. Precautions should be undertaken to protect the emulsion against degradation caused by light, temperature, and freezing. These are the stability problems, other stability problems, and how to overcome the pharmaceutical emulsion. Then, the efficient emulsifying agents can be grouped into three categories: naturally occurring emulsifying agents, synthetic surfactants, and cation synthetic surfactants, and finally divided solids. First one, a naturally occurring emulsifying agents. This includes polysaccharides, semi-synthetic polysaccharides, sterol containing substances. First one is polysaccharides. In this preparation of an oral emulsion, acacia is proven as best emulsifying agent due to its ability to form a thick film at the oil-water interface, which provides a barrier against coalescence. But acacia is too sticky not to be used externally. Thus, the tragacan is used as a viscosity enhancer, which also prevents creaming. Starch, pectin, and carrageenan are some of the popularly used emulsifying agents in this category. This is the polysaccharides. Then some synthetic polysaccharides. Less viscous grades of the emulsifying agents like methyl cellulose and carboxymethyl cellulose are used for preparing oil in water emulsions. The third one is sterol containing substances. Beeswax, wool fat, and wool alcohols are some of the examples of emulsifying agent of this category, which forms water in oil emulsion. Then synthetic surfactants. These emulsifying agents are divided as follows based on their ionic properties: anionic, cationic, and non-ionic surfactants. First one is anionic surfactants. The surface active anions present in these organs, for that is anionic surfactants, provides them their surface active properties. This may be non-compliant with some of the organic and inorganic cations and with other large organic cations like cetrimide. These are oil and water emulsifying agents meant for topical applications and are effective in ionized forms. Emulsions comprising anionic surfactants are more stable at alkaline pH. Few popular examples of pharmaceutically used anionic surfactants are alkali metal ammonium soaps such as sodium stearate, soaps of divalent and trivalent metals such as calcium oleate, amine soaps such as ethanol amine oleate, alkyl sulfate such as sodium lauryl sulfate, then cationic surfactants, second one. Since these quaternary ammonium compounds comprise of surfactant cations, they show sensitivity towards anionic surfactants and drugs. These are employed for preparing oil and water type emulsion intended for topical use. These surfactants are also more effective in their ionized forms and their emulsions are more stable at acidic pH. Few cationic surfactants like cetrimide, benzalkonium chloride possess antimicrobial activity. The third one is non-ionic surfactants. These synthetic agents, constituting the largest group of surfactants, are required for producing both oil in water or water in oil emulsions for external as well as internal applications. These surfactants are compliant with anionic and cationic substances. These remain unaffected to pH change. The emulsions formed depend on the balance between hydrophilic and lipophilic groups provided by HLB number, which means hydrophilic lipophilic balance number. Glycol esters, glycerol esters, macrogyl ethers, sorbitan esters, and polysorbates are few of the popular examples of non-ionic surfactants. In finely divided solids, a coherent film is formed when the finely divided solid particles are absorbed at the oil water interface. This coherent film prevents the dispersed globules to coalescence. A water in oil emulsion is produced when the solid particles become wet with oil. It contrasts an oil in water emulsion results when the particle become wet with water. In comparison to the emulsions formed by naturally occurring emulsifying agents, the emulsions formed by finely divided solids of better stability and are less susceptible to microbial contamination. Bentonite, aluminum magnesium silicate, and colloidal silicon dioxide are few popular examples in this regard. Colloidal aluminum and magnesium hydroxide are meant for internal preparations. For example, magnesium hydroxide is used to stabilize the liquid paraffin and magnesium hydroxide oral emulsion. Thank you.